If we were to understand how biology works, there was so much we could do to make everyone's life better. Today, we don't have genetic agency. Through all of our history of human civilization, we haven't been able to take action at the DNA level. Now we can understand what is needed, but we still can't take action because the therapies don't exist. But I like the term genetic agency because it really indicates that patients are going to be the ones who make this decision. We should be doing everything we can to give them more options so they can decide for themselves what to do. If we're going to realize the potential of genetic medicine, we need better technologies.
Welcome, Eric Kelsic, the CEO of Dyno Therapeutics, to the A16Z podcast. Thanks for being here.
Excited to be here. Thank you, Jorge.
So we have been investors in Dyno for some time, but I think one good place to start is to talk about what Dyno Therapeutics does and why it's important for genetic medicine.
Okay. Well, we are a genetic technologies company, and we partner with other therapeutic developers to make gene therapies. And in particular, we help our partners to make better medicines by solving the challenge of delivering their therapeutic DNA into the body where it's going to be active. So that could be to the brain, say to every neuron, could be to the muscle, to the heart, could be to the eye. And over time, we want to be able to reach every cell in your body, every organ, so that they can all benefit from genetic therapy.
And why is Dyno necessary?
Well, we really think that genetic medicine has huge promise, but if we're going to realize the potential of genetic medicine, we need better technologies. So the idea is very simple. We're all genetically programmed through our genomes to work in a certain way. But with a genetic disease, sometimes there's a change in the genome. Maybe you inherited it from your parents, or maybe it spontaneously arose. And those changes can sometimes cause problems. So to address the root cause of the disease, you really need to take action at the genetic level by adding back in a protein or another gene that replaces something that might be missing, or by making a correction in a way that puts your cells in a healthier state. That's the field of genetic medicine, and it's been a project for many, many decades. And over time, we've begun to understand what's truly possible. We've been able to sequence the entire human genome, which now means we can diagnose thousands of genetic diseases that before we didn't know what was going on. Now we know. Now actually you can get that diagnosis pretty quickly and pretty cheaply when we already understand the cause of the disease. It could be under $1,000 to sequence your genome and to get the answer. But that doesn't mean that once you know what's causing the problem that you can immediately fix it. So there needs to be a way to treat these genetic diseases, and that's the field of gene therapy. But for the past 40 years, we've had a really hard time delivering genetic medicines into your body. So that's the challenge of gene delivery. Dyno, as a genetic technologies company, we're focused on solving these types of challenges, and we've actually focused on delivery now for more than 10 years because it's really been the limiting thing for that's preventing progress in this field. And the reason why Dyno exists is because we really thought there needs to be a company that's focused on solving these big problems for everyone. So we're a platform. We're not actually developing genetic medicines ourselves because we think if we solve this problem, everyone's going to want to use these technologies, and we want to share them as broadly as we can so that there's even more impact and even more patients are going to benefit. So really a partner helping these other genetic medicine companies to develop even better medicines. And we provide the delivery technology, which is this protein shell called a capsid that takes our partner's DNA genome, which is the therapeutic part of the drug, delivers it into the body. And every one of our partners has a different DNA sequence. That's called the payload of the gene therapy. So one payload might be adding in a new protein with to restore a function of something that's missing. Another payload might be making an edit to a patient's genome or changing their RNA sequence or somehow regulating the expression of genes in a way that puts cells in a healthier state. So many different things you can do with genetic medicine on the payload side, but all of them need this delivery technology. So we want it to be a platform because rather than doing all these things ourselves, if we just focus on this one problem or a few of these problems that are really, really challenging, everyone's going to benefit. And ultimately, that's going to create a future in which genetic medicine, there's many more options available for these genetic diseases, which today many or most of them have no good treatment options. And hopefully in the future, genetic medicine will be something that everyone benefits from, maybe multiple times in their lifetime.
That's great. So you've coined a different term that you call genetic agency. What is genetic agency as it relates to a genetic technology or a genetic medicine?
So
help us unpack that.
Yeah, genetic agency, what it means is the ability to make informed decisions, to take action at a genetic level, the DNA or the RNA level, to live a healthier life. So it's a very simple idea, but I wanted to find a way to frame where we're going, the goals for all this work in genetic medicine in a very positive way. So genetic agency is essentially what everyone wants. And the rule for entrepreneurship is you want to make something that people want. We want the ability to take action. So you can imagine if you're a parent and your child has something that you feel is not quite right, and then eventually it becomes very clear there's a problem, but you don't know what the cause is. Well, then you go through this process of going to doctors, trying to understand what might be happening. Ultimately, you get a diagnosis, and when it's a genetic disease, the diagnosis can be very clear. Like, your child has this mutation. But what's really sad is today, when that happens, in many cases, the diagnosis is clear, but there's no treatment that's available. So while we understand the disease, there's nothing you can do. And many of these diseases are very serious, even very, very deadly. And that's really tragic that there's an understanding of what the problem is, but no ability to take action. So genetic medicine is a very generic sounding idea, but genetic agency just means the ability to take action at the DNA level. And when there's no treatment that exists today, we need to find a way to make it easier to develop those medicines. Could be through technologies like delivery that solve part of the problem, could be through technologies like AI that could automate some of these steps to make it faster or cheaper to develop genetic medicines. That's going to be a step towards genetic agency. In many cases, when a patient or a family member has a diagnosis like this, they see there's no treatment options available, and they want to take action, so they actually become therapeutic developers. They might start a company, or they might start a foundation to fund research, which then starts companies. This is happening more and more over time.
Yeah, and there are so many examples of that, and it's heartbreaking how much effort and work goes into finding a therapy when nothing exists.
That's right. And so I really wanted Dyno to be the genetic agency technology company that we're focused on the technologies that are going to make this even easier, even faster, even cheaper for all the genetic medicine developers who already have so many other challenges. If we help them with genetic technologies, that's going to be our way of having an impact in the space. And then the other reason why I think genetic agency is a really important term is that these therapies today, they're not perfect. They have challenges. They have risks. Even as you mentioned, there's been some deaths from gene therapy, but it's really important to think about all the risks and all the challenges as a big picture. So you have to contrast a therapy which may be experimental, the efficacy may be unknown, there might even be some risk of death, but you have to contrast that against a disease which is maybe already deadly, certainly deadly, and could be very urgent, or it could be that over 10, 20 years, everyone with this disease dies. And so patients are really looking for something that they can do. And it's really hard to say whether anyone should be taking a genetic medicine now, or waiting for a better one to be developed, or focusing their efforts on developing one themselves. But I like the term genetic agency because it really indicates that patients are going to be the ones who make this decision. And we should be doing everything we can to give them more options so they can decide for themselves what to do. As opposed to, really focusing only on the technology and saying these technologies are good or these ones need to be better. And in that way, maybe waiting to develop better medicines or delaying or slowing down the development of genetic medicines, when from the patient's perspective, that's not what they want. They want more options, even if they are not perfect.
Yep.
And then over time, more and more options that are really, really good. So the only way in which we can do that is by really democratizing the development of these medicines, making it so that there's competition between all the therapeutic developers to make the very best medicine, and then to make the one that's easiest to deliver, most accessible, maybe even most affordable over time. And that's the way in which we're going to really realize the full potential of genetic medicine, is through genetic agency.
So genetic agency is your vision to bring technology to bear to enable patients and their families and the relevant stakeholders to take action, to intervene in the face of urgency.
And it's really to highlight that today we don't have genetic agency. Through all of our kind of the history of human civilization, we haven't been able to take action at the DNA level. Now we can understand what is needed, but we still can't take action because the therapies don't exist. So, once we develop the technology to do this, and then patients are in the driver's seat, they can decide what they're going to do, where they're going to, take their life. They're making these decisions about how they can live healthier, and they have these options available. That's just going to really change how we think about ourselves and really create a really optimistic vision for the future, where we can solve these big problems that exist, but then ultimately bring this to a much broader audience. And the reason why I think that's really critical is for rare disease or ultra rare disease. It doesn't really matter what the disease is. The cost to develop the therapies are very similar. They're really complex. They're really expensive. So today, a lot of the focus in industry is on diseases which have larger patient populations, where once you invest in the development of that treatment, there's going to be enough patients that you can help, that you can recoup that investment and then use that to make even better therapies. But it means that there's another kind of segment of the market, these rare and ultra rare diseases, where it hasn't made economic sense to invest in developing those therapies. So today, those are often being worked on by nonprofits, foundations, even patients themselves who find that unacceptable. But it's because the economics don't work out to make this attractive for most companies.
Yeah,
but you can imagine
few,
if,
so to speak,
if gene therapy was something that everyone could benefit from, well, now there's this massive addressable market. And then you can bring economies of scale to the development of technologies, so that, if we can make the treatment of a super common disease like obesity even cheaper, or other things that might affect everyone as we age, that's also going to benefit the rare and the ultra rare disease patients, because they all use the same underlying platform technologies. All these therapies need delivery. All these therapies need different ways of changing the genome or adding a new function or repressing a function. So because fundamentally we all share the same genetics, because all these genetic machines work in similar ways, by making gene therapy a really big market, we can also make the development of new therapies even more affordable. So I think that there's also a lot of benefit of helping share that vision of genetic agency, not just with rare and ultra rare disease patients, or even not just with genetic disease patients. It's really something that, because we're all built through genetics, that it can help all of us live a healthier life. And as the therapies get even more effective and even safer, then you might not think twice about taking one or multiple gene therapies at some point in your life to live healthier. The same way that many of us have had some form of surgery, at certain points in our life. For me personally, like thinking about this idea of genetic agency, it's really grown on me over time. It's changed my perspective, where a lot of things that I took for granted before were always going to be the same. Now that I understand geneticology might someday be something that everyone uses, now I realize actually a lot of the decisions that I'm doing, maybe there's a genetic component, where if there was a technology or product available that I'd want to use, then I would think about that alongside all these other options as well.
Yeah, we might get to a point where interventional genetics is like a field.
Exactly.
So just to pick up on a couple of those threads. So first of all, if we had a magical ability to deliver any payload to any specific, cell or tissue in the body today, if we had that ability, have we gotten, has the industry, has technology gotten sufficiently developed where we could treat any genetic disease if we knew the mutation?
I think for almost every genetic disease, if we could deliver a therapeutic sequence to the right places, there'd be something we could do. There'd be something we could try, and in most cases, we could fix the root cause of the disease, so long as we understand what's going on, what's the problem. It might even be that in some cases, we don't understand everything that's going on, but we know the right way to intervene in a way that would still put cells in a healthier state. So there might be many different genetic mutations that all have the same ultimate kind of outcome. And if we intervene at a later point in the way that the cells are functioning, then we can kind of fix all of the problems just by a later intervention.
Right.
So
just to pick at that, right now where we stand today, we have gotten sufficiently advanced in genetic medicines, whether they're gene therapies or gene editors, correcting errors in DNA or replacing or augmenting genes. But today, the rate limiting step is the delivery piece, which is the insight you had behind saying, like, we want to be in the genetic delivery technology business.
That's great. Yeah. And I'll give you an example. So for example, actually just this week, there was a gene therapy company, uniQure, which had some really exciting new data from their latest gene therapy trial. So this is for a disease called Huntington's disease, which over time causes patients to lose function in their brain. And up until now, there was no way to treat this disease, to treat the root cause of the problem, which is this toxic protein which accumulates in your neurons.
And it's a devastating
a devastating disease. So uniQure created a gene therapy, and they use a strategy in which downregulates this expressed gene. And that can, but by reducing the amount of this toxic gene, they can treat the root cause of the disease and actually delay the progression of the disease by what they reported was 75%. And just to put that in basic terms, that might mean for a patient who was thinking about 5 years of health that they could enjoy, that could be extended to 20 years. So it's a really meaningful difference. And this is just the beginning, because this is the first time we've been able to treat the root cause of Huntington's disease. So now you can imagine that if this works, maybe we can make even better therapies that would extend health for even longer. Or maybe we can make even better therapies that would be even easier to deliver, or even cheaper to produce and then to administer to patients all around the world. For example, in this case, with the uniQure product, it's a direct injection into the brain. So it actually requires neurosurgery to get the gene therapy to the right places where it's going to be effective, which you can imagine is a very complicated thing. And it's going to be hard to do that for all the patients who need it all across the world. But in a next generation gene therapy, maybe we could deliver it through the blood, cross the blood-brain barrier, and then get into cells all across the brain. So that would have the benefit of potentially even more
an injection.
An injection, yeah. So it's kind of a really simple way to administer the gene therapy. That could be beneficial because it might even be more effective. You'd be reaching more cells all across the brain, so the treatment outcome might be healthier life for much, much longer. And it would also be much easier to deliver, so you could do this in more locations that wouldn't require neurosurgery. And if you could be super efficient, then it might even bring the cost down very dramatically. So these are the reasons why new technologies are still worth investing in. Even when we understand the disease and we know how to intervene, there's still going to be more patients who are benefiting, or more benefit that comes from further advancements in the technology.
And that's the opportunity, is to advance the technology so we can go and we can reach more cells, we can reach more tissues, and we can therefore treat more diseases with these advanced payloads. As you alluded to, your focus is not on developing your own gene therapies, but on enabling delivery of other other developers' payloads or cargo. I know you hate when I say this, but effectively you're acting as like FedEx for genetic medicine, where your job is to deliver everyone else's packages.
No, that's a good analogy.
So why do they want to call FedEx? Why don't they deliver their own packages?
Well, historically, that's what happened. And before Dyno, everyone had this problem of needing to find some way to deliver a genetic medicine to the body. But it's so complex to develop any genetic medicine. So not only do you have to solve delivery, but then you need to design the payload, you need a way to manufacture it, you need a way to test it across all these different systems before you go and do your human clinical trial. So because there's so much complexity, it's been really slow to develop these medicines. Or even if they've been successful, it's been really expensive. So the idea behind Dyno was, let's take some of these challenges, the ones that are shared across all these different medicines, and then we started with the first one, which was delivery. Let's take that challenge that everyone shares and really just focus on solving that for everyone. And with that focus, let's make it easier for our partners to focus where their expertise is best. And they can just trust when it comes to delivery that Dyno will take care of it for them. We'll give them the best technology today that will deliver on this promise. And when they need a better delivery technology, say for their next generation therapy or for the next product they want to create, they'll come back to us and maybe we'll have something even better by that time. So it's really, as the field grows, the specialization of technology developers and therapeutic developers will benefit everyone because we'll be focusing on our respective areas of strength.
Got it. Great. And so you mentioned partners. So today, where we stand in 2025, who is calling Dyno for delivery?
Some of our key partners are Roche and Astellas. With Roche, we're partnered to develop delivery to the brain. And with Astellas, we're partnered to develop delivery to the muscle, both skeletal muscle and heart muscle. Those are two organs which there's many different diseases that are impacted, and they're very difficult delivery challenges because you can imagine the muscle, it's all over your body. So there's many, many cells, many different regions that you need to treat. And in the brain, you want to reach all these different regions, and they're all slightly different, and you want to reach all the cells, all neurons, and even other cells like astrocytes. So in these cases, people have been trying to develop genetic medicines for many decades, but the delivery challenges has really limited the progress. It hasn't been as fast or as much as we would all like. With Roche, we actually are on our second partnership. So the first one started back in 2020 and was also focused in the CNS. And earlier this year, Roche licensed the first capsid from Dyno for a neuroscience indication.
Fantastic.
So that's now moving in their hands towards clinical trials. And now we're focused on our second partnership, which is much broader, but also in the CNS.
Fantastic. Okay, so let's talk about delivery technology. You described it as a shell a few minutes ago.
This capsid.
This capsid. So what is a capsid? Where does it come from? We can help our viewers understand what they look like because you're wearing one on your chest. So tell us what a capsid is.
Yeah, so capsid, this is the one that we focus on. It's this icosahedron. It's a protein shell. You can think of it just like a really amazing protein machine. So it does exactly what's needed to deliver a genetic medicine. And the way in which we've created them is we've looked in nature, where is this happening naturally, and learned from that, and then taken the best things that exist in nature, which are these capsids that come from an adeno-associated virus. You can imagine viruses naturally want to deliver their genomes into your body. So AAV is one of the smallest, one of the safest of these natural viruses. Looking at how it works, we found the capsid proteins, and we've taken them out and then started to modify them to make them even more effective as therapeutic tools. And we do that by changing the sequence of the capsid protein. So every protein has a amino acid sequence. And in this case, with the capsid protein of AAV, it's just over 700 letters long. So by changing those letters, say changing an A to an S or a T to a C,
these are amino acid
these
changes.
are different amino acid changes. We can alter the way in which the protein works. We want to alter it in ways that are beneficial therapeutically. So, for example, we want to make the capsid bind to cells that will enable it to get into the brain, crossing the blood-brain barrier, or enter into neurons without breaking any of the natural functions. Because what capsids are already very good at is once they're inside of cells, they can then traffic into the nucleus, which is where your genome exists. And then when they're in the nucleus, they can express their genetic payload, which is what makes the medicine active. So we want to maintain all those kind of natural good properties of the capsids while adding new functions that make them better therapeutically. And that's going to make them more effective, which is the most important thing. You need to reach enough of the cells for the genetic medicine to be useful. But we also want to make them even safer. And we can do that by making them super specific to the target cells. So if we want to treat the brain, we want to go only into the brain and avoiding other organs in your body, like the muscle or especially the liver. Most natural capsids are almost all going to the liver because it's the easiest place to get to in your body when you're going into the blood.
It's the body's filter.
It's
right?
the body's filter, exactly. Yeah. So with a natural AAV capsid, maybe 1,000 times more would be going to the liver than to the brain. And we've been able to actually invert that ratio. So now we're able to get more delivery into the brain than the liver or anywhere else. So it's over time really more fair to not call these viral delivery, but to say this is a capsid. It's a delivery technology, and it's becoming unlike anything that exists in nature because we're modifying it to be useful therapeutically. And that's going to continue over time. Similarly, we can change them to go into the muscle, or the other area that Dyno has focused on has been delivery into the eye. These are all great targets for gene therapy because these cells are not dividing, which means once you get the genetic sequence in, those cells will be there for your entire lifetime. Which
the payload will just
basically mean it's an effective cure for many, many years, many decades, maybe even a lifetime cure.
So going back to you keep emphasizing the phrase delivery technology.
Yes.
We have this protein capsid shell that originally comes from virus that's been modified to have therapeutic qualities, the ones you were describing. And you describe that modification happening via changes you make to the amino acid sequence of the capsid. How do we know what changes to make? How do you know what to design?
Well, we don't really know. The space of all the possible things we could do is just like, writing a book. You can imagine there's so many different words you could choose. The space of all possible books, it's infinitely large. So we need some way of making smart choices. And historically, proteins have been so complex that we didn't really know what to do. So the only way to make progress was to try a bunch of things and then choose the ones that work and continue to improve upon those. That's no different from how it works in nature with kind of natural evolution. This is just applying that in a more directed way, which is called directed evolution. And you can think of it like a needle in a haystack approach. You're trying all these different things, one or a small number work, and then you build on that success. And it takes a long time. Evolution took billions of years. And we don't have that much time ourselves. So we want to find a way to speed it up. And the kind of innovation that's powering Dyno's platform was a few technologies. One is that we now have the ability to gather a lot more data. So when we're doing this needle in the haystack approach, we can understand for all the things that didn't work, well, what happened? And we can learn from that. And to put this in more like practical terms, we can make many different capsid proteins, say hundreds of thousands or even millions of different capsid proteins. Each one differs by a few amino acids. It's programmed in a different way. But each of those capsids will have a different function. So they might go to different places in your body. Some of them won't work. Actually, most of them, if it's a needle in a haystack approach, are going to be broken.
They're just not functional.
They're just not working.
Yep.
So we make all these different changes And then we also include within that capsid a DNA sequence, which is called a barcode. And that barcode is a way of knowing what's going on with this capsid. And wherever the capsid goes, the DNA goes with it. And then we can sequence, which means we can read out those DNA barcodes with DNA sequencing technology. And we can then know which of the capsids went over here, say to your brain, which then went down here to the liver, which then went to the muscle. And then look back at the sequences that cause those changes and find patterns. And we can notice, oh, these changes in this part of the protein made it go better to the brain, or these changes in this part of the protein completely broke it. Don't try that again. And we learn from that how to make a better capsid protein. You can imagine we could do this ourselves, manually going over all the data, but if there's millions of different data points, that's also going to take a really long time. So the other technology that's really key to our platform is using machine learning and artificial intelligence to automate that process of looking at what happened, finding patterns, coming up with ideas of things that would be worth trying, double checking them, and then taking all of our best ideas and putting them into the next experiment. So that's the AI platform that we've built over time, is automating the analysis of all this data across all the measurements we take from all of our different experiments. Some of them are in the lab, testing whether the capsids bind to human proteins that will enable us to get into the brain. Others are done in animals, some in mice, but mostly in cyno monkeys, because that's the most relevant animal model for knowing what's going to translate effectively to humans. We gather all this data, we put it into machine learning models, and then we query those models to design the best sequence.
So originally, capsids evolved by nature. Dyno capsids are designed with AI.
That's exactly right.
And you've been at this for 10 years, right?
Yeah.
And so you came at this very early on. You were one of the, I would say, one of the early pioneers of using AI to design, in this case, viral vectors, or let's call it a complex biologic. How did you come up with this as the idea that you wanted to work on, that you wanted to spend your career on? Because I can't imagine, as a kid, you said, I want to design viral vectors using AI.
Yeah, it wasn't on my radar growing up.
You have a background in physics at Caltech. You were at the George Church Lab at Harvard. Like, tell me a bit about how that journey gets you to where you are.
The story of how I came to focus in this area and why I find it so exciting and still do. I always wanted to understand how things work. So that's what led me to physics. And then from doing physics research, what I realized was actually the thing that we have the most mystery about is ourselves. Like, how do we work? How does biology work? So I was drawn into biology because there's so much that we don't know. And yet, if we were to understand how biology works, there was so much we could do to make everyone's life better. So it's the combination of the mystery and the puzzles that you can solve by doing basic research, but then also the potential for really clear and direct impact on all of our lives. And then kind of the same thing drew me from academia into industry, and especially into startups, was wanting to see these results applied in a really impactful way. So for my PhD, I worked to find what was the types of problems that I'd be really good at solving. And that's what drew me to these areas where you could gather a lot of data in just a single experiment. And when I did that, I thought this would be really useful for protein engineering. And if so, what's the area that this could be applied in a way that would be really relevant for human health? So I went to work with George Church as my postdoc to figure out what was the best way to bring this technology into industry, and especially into therapeutic medicine. That's what led me to genetic medicine. That's what led me to understand that all gene therapies have this delivery challenge that is limiting progress. And so we have this new powerful protein engineering technology that's coming. Why don't we focus on where it's going to have the most impact and really develop the tools that will then be possible to apply in many other areas. So we focus on solving delivery because that's a challenge that if we solve it, makes genetic medicine much more valuable. But we've also developed a whole workflow for how to do AI-powered protein engineering that now we can apply and also share with our partners to help them design better payloads, or maybe even to help everyone design better biologic medicines. The idea behind that was that we can't do everything for everyone all the time. So let's just focus on the problems that we can make the most progress on. And sometimes it's better to focus on the hardest problems first, because that also teaches you how to develop the technology. So we were very early in understanding that there is this big opportunity coming with new data generation capabilities. And that led us to understand, well, there's also a new opportunity with AI to analyze that data. And 10 years ago, there was very little going on. Now, everyone sees that opportunity. But we were very early there and developed some of the first generative approaches to protein design. And then over time have been adding in even more powerful models, including most recently these structural-based foundation scale models, which we apply to make the capsids better, but now we can also apply to make the payloads better. So these technologies ultimately are very general purpose, which is why we think about this all as one genetic medicines platform that's going to really enable a whole new way of treating disease, and ultimately enable all of us to be able to take action at the genetic level in a way that wasn't possible before.
Yeah, and that's critically important, because you mentioned the example of Unicure with Huntington's disease. We talked about the fact that there are many genetic diseases that in theory could be treated with advanced payloads if only we could deliver the payloads to the right place. But there's also the other side of this, which is the current delivery technologies have their downsides. So there have been, if you go back and look in the news for gene therapy, there have been deaths. Sarepta had a couple of deaths. A company called Capsids recently had a death in a clinical trial. So the need is also real to come up with better technologies. Tell me a little bit about why these deaths happen, and why you think that a better viral vector is the right answer, a better capsid is the right answer, versus a different approach. So there are a lot of other approaches attempting to solve the delivery problem.
Yep.
And so, like, if I could completely abuse the Winston Churchill quote, where viral delivery is the worst. Viral capsid is the worst form of delivery, except for all the other ones that have been tried from time to time. Why do you continue to believe that the capsid is going to be the technology that solves the problem of delivery versus any of these other approaches, whether they're lipid nanoparticles or other non-viral approaches? Like, how do you know that we're not working on the Betamax of delivery?
Well, so I definitely agree with that that phrase, that capsids we have today, like, they're terrible, but they're the best thing we have. They're the best way to get it to the brain, into the muscle.
So you agree with Churchill?
Not other organs. So I definitely agree with that. But there is also this potential to make them much better. And we've really just started to apply these new, very powerful protein engineering technologies to make that better. And I think take a very pragmatic approach to this. There's really trying not to create distinctions where they don't need to be created. And I think what matters less for the future is where we're starting from, whether it's viral or non-viral delivery approaches, and more about where we're going. So where we're going is we want the ability to deliver to all the cells in any organ, or really across any location on the body. And we want to do that in a way that's targeted to those cells. We want to do it in a way that's super specific to those cells. And that's what's going to make the therapies very effective, very safe, very affordable. So whatever way enables those things is where we want to go.
That's the North Star.
And we've started in different places, but we're kind of converging in that direction, whether it's adding in functional parts to the delivery technologies to make them more specific, or to make them more targeted to a specific cell type, more efficient for delivering to those cells. All the ways that people are thinking to modify the delivery technologies we have today, they're all quite similar. They're using the natural kind of functions that biology offers us and making them genetic machines that are more effective at getting into the right places. So I'm sure that someday there will be some other technology that means AAV capsids are no longer the best thing. But maybe that will be done in several steps, starting from kind of a capsid-based approach, or maybe from another approach like an LNP. What we really want for our partners to do is to, trust that we will help them figure that out, and they can count on us to solve delivery for them. And then they can focus on the other huge challenges that exist for treating any specific disease, both how to intervene genetically, as well as how to manufacture, how to develop, how to commercialize that product. So I think that we're all actually working towards the same thing. And the whole challenge, though, is how to get there very quickly. And for me, starting with Capsids, because it was the best thing that we had today, plus also applying these very powerful genetic engineering technologies, still makes me very optimistic that that's going to be the path that gets us there the fastest, because it's already the best thing. And now we've found ways to make it hundreds or sometimes even thousands of times more efficient than when we were starting. And there's also a clear roadmap for how we can make these technologies even better. So as long as that looks promising, that's where Dyno is going to be focusing our effort. But as soon as something comes along that looks like it might be AAV, then we'll definitely take a look, and we'll add that into the platform.
Okay, so to realize that vision of genetic agency, as you're articulating, as you're describing it, is going to require you to bring together a bunch of stakeholders, whether they're scientists, technologies, patients, patient advocates, probably regulators, and you have an idea to do that. So talk a little bit about the conference that you're pulling together.
That's right. So I'm really excited that we're going to organize a conference to bring together all these folks who care about genetic agency, who want to make this future a reality sooner. It's called the Genetic Agency Technology Conference, GATC.
Clever.
GATC are the 4 nucleotides in DNA, so there's a nice ring to it. And we're going to have the first one in Boston in November. And it's very much a community building conference. So we want to bring together genetic medicine developers, but also technology developers. So both gene therapy, cell therapy, AI for protein design, even AI for automating some of the work that goes into therapeutic development. And then importantly, patients, their families, patient advocates, ultimately the beneficiaries of the technology. Like I said, for technology development, the most important thing is you want to make something that people want. It's not worth it if you develop a technology and once you've made it, it's not the right problem that you're solving, or no one wants to use it. So as we bring together all the developers of the technologies, I wanted there to also be the patients who really understand their disease. And many of these patients, as I said, they've become therapeutic developers. So they also understand the challenges. Bringing together these different groups early on can help us develop the technology that will make it much easier to develop genetic medicines in the future. And then we also need others to make this possible. So we want to bring in folks who understand kind of the commercialization of these technologies, both in industry and more broadly in a nonprofit context, and regulators who can help us, do the right things when it comes to hitting the target the first time for the development of the therapy. Maybe also change the way in which we regulate genetic medicines to make it easier, to make it faster, and ultimately to enable patients to make a more informed decision about when is the right time to take a genetic medicine, knowing that lack of action can be a really terrible thing. So we want to create more options that make it possible for patients to make that right decision for themselves, and in a much more affordable way.
Fantastic. So when you talk about make something people want, what if somebody wants to attend this conference? What do they need to do?
So you can go search for it online, Dyno, DYNO, at GATC. And it's a small group, so it's you can apply to attend, but we really want to bring together a broad group of stakeholders. And anyone who can make the case that this is something that I really want to contribute to, or this is something that I really want, I really need, or I'm going to benefit from meeting all the people who are going to be there, is very welcome to attend. We have an application on the website that's really simple to fill out. It's November 11 in Boston. And there's going to be all sorts of different people there. So both gene therapy developers from many different companies, including our partner Roche, and then also patients and patient advocates. I'm really excited to have Victoria Gray, who is the first patient who treated their own cells to cure themselves of sickle cell disease with their genome editing therapy. Victoria is really amazing, and she's got an amazing story to tell. And now she's an advocate for genetic medicines of this type because it's really changed her life. And I think that would be a really inspiring story that many people will appreciate hearing, as well as enabling us to talk about the challenges that exist today and how we need to find some way to overcome them to make genetic medicine a better technology.
Wonderful. Well, I hope you approve my application. I'd love to be there. And then just to end on a personal note, your vision for genetic agency and making this technology accessible. You were kind enough to host me and my 4 children when we were visiting in Boston. And I wouldn't say the Dyno headquarters is exactly Willy Wonka and the Chocolate Factory, but it's really cool. Like, it's a really cool place. It's really cool and inspiring to see what you're doing there. And I just want you to know, from me, number one, the kids were really inspired by that. They all have Dyno stickers on their laptops now. And number two, all that stuff was broken before we got there. Just to be very clear.
Well, you're always welcome to come. And I think that genetic technology is just such an amazing time to be in the space. So, for any science lovers, it's a good time to get into the field. And for any, kids, there's just so much potential to learn really cool stuff, like understand how we work in terms of our bodies, but then also solve all these mysteries that will enable us to use that knowledge to live in a healthier way. So that's what went into the design of our space, and I'm glad your kids had that experience.
Yeah, kids learn to code biology.
Yeah.
Eric, thank you so much for joining us today on the podcast.
Thank you, Jorge.
Really a pleasure to have you here.
It was great to be here. Thank you so much.